Uh, Thanks, Chiara. So let me then invite the first speaker, Andrew Reed. Andrew has been studying adaptation of disease-causing organisms to drugs, vaccines, and competitors for over 30 years, particularly the organisms causing malaria, Marek's disease, myxomatosis, and hospital-acquired sepsis. The title of his talk is Experimental Studies of Pathogen Adaptation to Host Immunity and Vaccination. So now it's with you, Andrew.
Thanks very much. Somebody going to project the talk? There we go. Thanks very much for the invitation to be here. It's very, very exciting. I am not going to talk about any empirical work going on in my lab with SARS at the moment. We're doing work on serology and wastewater, but I am going to talk about work that we've done in the past on studying pathogen adaptation to host immunity and vaccination. And just to get the wording clear here, I want to make sure that we're on the same page. I'm talking about host adaptation following host spillover. So that's the evolved ability to get better in a, sorry, I should say better in a new host. And then vaccine resistance is the evolved ability to replicate better in a vaccinated host. So the evolutionary process that leads to adaptation that leads to improved performance in either a new type of host or a vaccinated host. And vaccine resistance can be caused by a variety of different mechanisms, most obviously target sites, so that the antigen being targeted can evolve to be less well recognized by, for example, antibodies. But it can also involve immunosuppression, life history traits that allow it to replicate faster, get into cells better, and do things earlier and so forth. And I'm going to briefly talk about work we've done in the past on malaria, Marek's disease, and myxomatosis, and the quicker and quicker on each of those topics as we go forward, and then conclude with a few points about vaccine resistance in humans and agriculture. So this is data from an old experiment. We used to do lots of these sorts of things in the first decade of this century. We would passage malaria parasites through mice. So the species we use, Shabodi, is comes from a thicket rat in Central Africa and has been mouse adapted. And it's well known that the more you passage it through mice, the better it gets at dealing with mice. And so we used to do a lot of experiments with serially passage through mice with syringe to syringe. And depending on the genotype and the clone or whatever was going on, would be doing this for 10 or 30 passages. And this is one data point. I should just say too, that starts with a single parasite. So it's a clonal process of amplification through the different hosts. And this is a single experiment with one of these lines. The ancestral line is how the parasite was performing in the pre-passaged parasite was performing, and the derived line is what the performance was like after the serial passage. And you can see that it sort of grows maybe the same to start with, but it's doing better. The derived one is doing better later on in the infection when it's less well cleared by the host. If you take those exact same parasites, the ancestor and the derived one, and you put them into a vaccinated host, then this is what you get. And this is the vaccine here is one of the candidate human antigens, malaria vaccines, and but modified for this particular species of parasite. It's a commercial adjuvant is added, and this is the performance. And so you can see that the parasites that have been passaged through the naive mice, so through the new species of host that have got doing better at the parasite at the new host, are also much better at growing and replicating in a vaccinated mouse. So even though they've never evolved in a vaccinated host, this is the first time they've seen the vaccine. The process of having got better at being in a mouse has led to improved performance in the vaccinated host compared to the ancestor. We've seen this many times in many different settings. That led Vicky Barclay to do a heroic experiment where we did experimental evolution through mice that had been vaccinated and mice that had not been vaccinated. So you take a single parasite and evolve it through time in these different lineages. The control here is just adjuvant, and the vaccination, the V lines are going through the adjuvant plus the protein, recombinant protein treatments. And you can see, just looking at this, this is the lineage that we tried to maintain 5 lineages, all points. You can see that we had the red dots are when they die out because there's not enough parasites to transfer onto the next host. And so you can see here that we had no problems keeping the C lines going, but we had a lot of trouble getting the V lines, keeping them going, and most of them died out most of the time to start with. But round about passage 10 or 11, they started getting quite good at these vaccinated hosts, meaning that we were able to find enough of them to passage on to the next host. And so by generation 20, we had some lines that we were calling vaccine adapted. This is the process that what happened through time here. So this is the parasite densities at the day we transferred them over. Each of the points is a mouse. The blue points are the mice that are not vaccinated, and the red points are the points where there was the mice were vaccinated. And so you can see at the start, serial passage number 1 and 2, the vaccine was really suppressing the parasite densities at the day of transmission, which was a week. But as things moved on, the parasites got better and better, and they were hitting higher and higher densities. So by passage 21, they were achieving parasite densities in the vaccinated mice that were in line with what we were getting in the unvaccinated mice. So there's actually no difference at the end. So that means that after 20 of these serial passages, the vaccination was having no impact on the parasites in the sense that the anti-parasite protection had been erased. So the parasites got to the point where it was as if the mice were not vaccinated from a point of view of the parasites. And when you look at the gene involved, the protein involved, there was no evolution at the target site. So the evolution was happening somewhere else in the genome. And importantly, even though the parasites were doing just as well, the anti-disease protection was maintained. So the mice were still healthier if they had been vaccinated, even at the end, than if they were not vaccinated. So the parasites got better and better, but they did not make the mice as sick as if they hadn't been vaccinated. Now, I believe this is the only experimental evolution anybody's done for vaccine resistance. Although if anybody knows another example, I'd be delighted to hear it. Very large experiment done by Vicky. She then took parasites at day 10, at passage 10 and passage 20, and put them into naive mice, so mice that were not vaccinated. And you can see there's a lot of noise in there. The mice were making the parasites that evolved in the vaccinated mice were causing greater anemia and were growing to higher densities in the non-vaccinated mice. So not only had they got better at vaccinated mice, they'd also got better at unvaccinated mice, and they made them sicker. So from that point of view, then we concluded, and that's a subset of the experiments that we've done in that serial passage in unvaccinated hosts leads to rapid adaptation to novel hosts. So when you put a parasite into a new type of host, it rapidly adapts to that novel host. Others have seen that in many different systems. Serial passage in unvaccinated hosts leads to vaccine resistance. So a side effect, a correlated response to adaptation to the novel host, was that the strains that evolved were better at dealing with vaccinated hosts. That was also the case when the parasites evolved through vaccinated hosts. So you get vaccine resistance would arise as a correlated effect of adaptation to novel host as well as adaptation to vaccinated hosts. And in all of these cases, the vaccine resistant strains cause more severe disease in unvaccinated hosts. So it rapidly evolves in the system, vaccine resistance. It's correlated response to adaptation to novel hosts. And I want to re-emphasize, this is not due to the evolution of the target locus. So in the SARS case, for example, most of the vaccines are targeting, all of the vaccines are targeting the spike protein. The equivalent here was the AMA1 protein, and the evolution that mattered here did not take place in that locus. I now want to talk briefly about Marek's disease. This is a chicken virus, DNA virus, that has been vaccinated against since the 1970s, but 2 generations of vaccine failed in the face of virus evolution and independently in different parts of the world. And so vaccine resistance evolved at least twice in the, well, at least to 2 vaccines and in different parts of the world. So we've been interested in this process of vaccine resistance for some time now in this system, where the vaccines did have to be replaced because of the economic losses due to the evolved virus strains. This is what the vaccine resistant strains look like. This is on the left is a vaccine sensitive virus strain, and you can see that when you inject them into chickens, the virus densities in the unvaccinated birds are much higher, several orders of magnitude higher than in the vaccinated birds. So the vaccine is suppressing replication. Whereas if you look at the vaccine resistant strain, you can see that it is actually growing very well in a vaccinated bird, and it's growing about as well as the sensitive strain in an unvaccinated host. These vaccine resistant strains are typically lethal in unvaccinated birds, and so you can see that in fact in this particular case, the strain killed everything by day 10 or so if the bird wasn't vaccinated, but vaccination allowed the bird to stay alive and the virus to replicate to high levels. So these vaccines are live attenuated vaccines, but they're very imperfect. They allow a lot of replication within the birds and a lot of onward transmission in the chicken sheds. And these are shed, the virus is shed as dust. And just to give you a flavor of that variation in vaccine susceptibility and resistance, these are 5 strains, the vertical panels, 5 different strains. The one on the left, the dotted lines on the top panels, those are the death rates in unvaccinated birds, and the solid lines in vaccinated birds. So the one on the left kills half the birds in a couple of months. The ones on the right kill all of the birds in 10 days. And they are starting to kill some of the vaccinated birds as well. And we've been measuring the viruses that are shed from these birds, and then you can calculate the cumulative viruses that are shed during the lifetime of these birds. And you can see that the one on the left, the strain on the left, is vaccine sensitive in the sense that the vaccination suppresses shedding. But as you go across to the right, you see more and more that the vaccinated ones are the ones where most of the shedding is happening. They're working very well in those cases. And so the performance of those strains is actually higher in the vaccinated birds than the performance of the ones on the left. And so there's variation that's evolved in the system for vaccine resistance, and that's associated with disease severity. And in our view, and I haven't got time to go through it, the reason for this is because the vaccines leak so much, there's so much transmission going on in these systems that there's lots of potential for evolution. And finally, I just want to mention myxomatosis, which of course is the canonical example of a species, a pathogen evolving very rapidly in response to spillover events. So it was released in Australia, spread, perhaps killed 100 million rabbits in the first year or so. And Frank Fenner was smart enough to realize that the virus might well evolve less virulence through time. So he started sticking the viruses into lab rabbits and showed that indeed the virulence did drop very rapidly. And he kept that work going until the 1970s. We picked it up again just recently in the last decade or so, and of course did all the sequencing of the more recent strains. But we also did Fenner-like experiments where we took strains from the 1990s and injected them into the standard lab rabbits. We also took Fenner's strains and injected them into the lab rabbits. And what we found was we could recapitulate Fenner's results, but we also found that the 1990s strains caused death in a completely different way. So on the left is the classic mutation, the classic myxomatosis. The rabbit's got puffy eyes, it's got big lesions and so forth. On the right, one of the 1990s strains just kills the animals at roughly the same time, but they die without any classic signs of myxomatosis. So they don't have any big lesions. And what happens is they bleed everywhere. They've got bacteria infiltrates through all sorts of organs and so forth, but there's absolutely no inflammatory response. So what the virus has done through the 80s and 90s has evolved the ability to massively immunosuppress the rabbits. You don't see this in the field because the rabbits themselves are evolving resistance as well. So the arms race has led to an escalation, which continues to this day. We've got some more from the strains now from the 2000s, and they too are massively immunosuppressive viruses. So the arms race between the host and the pathogen has led to very, very immunosuppressive viruses. And that's been the picture, I think, less well documented, but wherever people have done these pathogen transfer experiments, where people have put the pathogen from the field into a common garden host, it's always been an escalating arms race. So in these particular cases where that's been done, and this is ranging across, RNA viruses, DNA viruses, etc., different settings, has always been an escalation. So that the resistance that's evolved in the host has been met by increasing virulence in the pathogen. Now, I don't know whether host resistance in the wildlife setting has much to do with immunity in vaccine-induced immunity. I just want to make the general point that escalations of arms races, where the experiment's been done, seem to be the rule. Now, it might be that we only do the experiments where escalation might have taken place, but we don't have situations, I think, where things have evolved downwards and kept going downwards. And myxomatosis had initially evolved down, but then turned and went up again as the rabbits evolved resistance. And so just to conclude, Dave Kennedy and I have recently been looking a lot at the vaccine, in the pre-COVID era, looking at vaccine resistance in human and agricultural settings. And vaccine resistance evolves less often than drug resistance. But even when vaccine resistance has evolved, it's very interesting to us that vaccine-resistant strains haven't gone to fixation. There's always some degree of cross-protection, so that even when there is a strain that is less well prevented by vaccine-induced immunity, there's still some cross-protection, so the pathogen's not growing as well. And there's still anti-disease protection, often quite a lot of anti-disease protection, even if the pathogen itself is able to replicate better in the host. And here are 4 of the, I think, 10 or so cases that we looked at, where despite the vertical lines are when vaccines were introduced and new generation vaccines introduced. And despite the evolution in these systems, the pathogens themselves are obviously substantially less common than they were. And just to emphasize that, this is Marek's disease in one of our Pennsylvania study farms. And this is one of the farms itself where we've been following them for a couple of years, and that's the virus concentrations in the system. This is a vaccinated farm. There's virus around, honking amounts of virus transmitting all the time, and yet the birds are not sick. The vaccination is keeping the birds healthy until they go off to market. And so there is substantial gains to be had from the vaccine, even though there are vaccine-resistant strains spreading and circulating in the farm. If things get too bad, they use one of the 3rd-generation vaccines. Otherwise, they can maintain the system with the 1st-generation vaccine, even though there are viruses that are able to grow in those animals. So when you look at the harm caused by vaccine resistance in the cases where they're well documented, there are different mechanisms of resistance that appears, and there are changes in the disease burdens, but still the hosts are better off when they are vaccinated. There's always some anti-disease protection, often quite a lot. So, from this, what can we say about SARS-CoV-2? Well, I just want to emphasize that we don't know how much we can generalize here at all. And this is the paper that we published on the malaria vaccine. The editors and the reviewers made us state here that it was not just any old vaccine, but a blood stage. It was not just any old malaria, but it was Shibboleth. They asked us to put in a section on the caveats in the discussion and a whole box on evolutionary risk. And so I've got there in the box the sort of things we talked about, all of which made the reviewers in the journal happier that what we were talking about with vaccine resistance might not be a general phenomenon. And I think we just simply don't know here what generalizes and what doesn't. When I look at SARS-CoV-2, I see leaky vaccines, big open question as to how leaky they are. I see single target vaccines, and I see host adaptation, rampant amounts of host adaptation going on at the moment, which is generating at least some strains that seem to do better in, if not vaccinated hosts, certainly in vitro against antibodies generated by vaccinated hosts. So open questions are just how leaky are these vaccines, and what is leaking? Is it random viruses, or is selection happening when the vaccines are leaking? A lot of attention is being paid to the anti-spike evolution, the evolution in the spike protein, but there's evolution happening elsewhere in the virus as well. And it's a very interesting question as to how many other different phenotypes might be going on as well as antigenic escape. And I think one of the big questions is how disease severity impacts transmission, which I guess may be what Jess is going to talk about later. I still feel slightly relaxed about all of this because I do think the vaccine platforms are easily tweakable, and so quite exciting things can be done. And I just want to end on the note that because I've been getting quite a hard time recently from the anti-vaxxers and indeed some pretty irresponsible media outlets in America, none of the things we've ever done in the past give any reasons why you should withhold vaccines. It's always better off to be vaccinated no matter what evolutionary outcome happens. at least in all of the situations that we've studied so far. And with that, I'll stop. Thank you very much.
Thanks, Andrew, for the fascinating talk that you just gave us. So I don't see any questions in the Q&A chat, but I do have a couple questions for you. The first one is, what do you think that should be the observational and the experimental studies that we could think about in order to explore all the uncertainty with the COVID vaccine?
Yeah, well, the observational ones, I think, are, from my point of view, very obvious. We want to know, with some random sampling of vaccinees and non-vaccinees, what sort of viruses are in those people. So we know already that there are people who are vaccinated who are virus positive well after they should have mounted protection. So there is something going on. Now, is that random or not with respect to the virus genotype? That's not actually a terribly hard thing to work out. And I think that's the top priority. I'd also like to know a bit more about the diversity of immune repertoire that's being developed within people and between people. Is my antibody or T-cell repertoire the same as yours once you've been vaccinated? Because if there's diversity in the response amongst people, so different epitopes are being targeted by different people, then even if one person leaks, another person might kill that escape variant. So those are the sorts of things I'll be looking at in terms of the observational studies, and obviously the very hard question about whether the disease severity is greater for these evolved strains. That's also vitally important, though, as everybody I guess on this call knows, very hard to look at. In terms of the experimental options, they're actually quite limited at the moment, I think, because with all human pathogens, you have significant trouble doing the sorts of experiments that I've just been talking about. I do think it'll be very, very good to develop some coronavirus animal models that don't have to happen in a BSL-3. So mouse coronaviruses would be something I think that could be really well developed in this sort of space to give us some guidance as to the sorts of things that could happen under repeated transmission. Okay, thank you. So we have one more question, and we do have time for that. So Andy Dobson asks, super talk, and when the parasite evolves resistance to the vaccine in non-vaccinated hosts, are there any key difference between this resistance and that from strains passage through vaccinated hosts? Yeah, hey, Andy, that's a great question. When we did those experiments about a decade or so ago, it was way too expensive to do full genome sequences, so we haven't done that. All I can say is that they're identical at the target locus, because that didn't evolve, so the AMA1's the same. In terms of the broad life histories, our sense was that the ones that have evolved through the immunized hosts typically grow faster earlier, so they replicate more rapidly earlier on. And that might be something to do with the advantage that they're getting in the vaccinated host. Maybe they can stay ahead of the immunity as it's ramping up. And that gives them an advantage in the unvaccinated as well, of course. But that pressure might be stronger in the vaccinated hosts. But that's really speculation. And looking back on this now, it wasn't the reason we were doing these experiments, but it would be very interesting to do head-to-head competitions between the ones that are evolved to deal with the new host and the ones that have evolved to deal with the vaccinated new host. Okay, so thank you.